So in this video, we're going to begin discussing eukaryotic transcription, and we're going to start with the initiation and elongation phase. So in contrast to bacterial transcription, eukaryotes have a variety of different RNA polymerases that are going to focus on different types of promoters and certain types of gene sequences. So the main three that we're going to use to encode RNAs are RNA polymerase 1, 2, and 3. RNA polymerase 1 encodes rRNAs. Polymerase 2 encodes your mRNAs, so all of the RNAs that are eventually going to encode proteins. That is RNA polymerase 2. And then polymerase 3 is going to encode mostly your tRNAs and also one specific type of rRNA. So what we're going to focus on in this video is RNA polymerase 2, which is synthesizing our mRNA, because in terms of gene expression, when we talk about the central dogma, this is the one that is in that process of DNA, transcription, RNA, translation of proteins. So if we look at a type 2 promoter region, you see that there's this region around the -25 sequence called the TATA box, which has this consensus sequence TATAAA. That's kind of the consensus sequence within this promoter that's recognizable, that the cell's going to recognize and notice as, okay, this is a promoter region. We want transcription to start near here, and that's what's going to attract your polymerases and your machinery in order to start transcription. So of course, it's slightly upstream from your +1 transcriptional start site. And then of course, further upstream from the promoter is your regulatory sequences, your enhancers, your silencers, which might regulate the transcription of this gene. So contrary again to bacterial transcription, where we just had one RNA polymerase with a bunch of subunits, for basal transcription in eukaryotes, you have, to remind, basal transcription is kind of your normal rate of transcription. It's this baseline low rate of transcription. When we talk about basal transcription for an RNA polymerase 2 or a type 2 promoter, you have not only just your RNA polymerase, but you have a bunch of what are called general transcription factors. They're these proteins that have to also come into play to help kickstart your transcription process. So let's go through the list though in the order in which they come in. So the first one that's going to come is what's called transcription factor 2D. And this is responsible specifically for locating and recognizing that TATA box. It's going to find this TATA box and recognize this region as a promoter where we want to start transcription at. And then TF2B is going to be recruited, which is going to come to the promoter region, and that's going to help bring RNA polymerase 2 to the promoter region. So then, of course, RNA polymerase 2 is going to be brought in with TF2F, which kind of comes bound to RNA polymerase 2 and helps bring it along. And then TF2E is coming into play, which helps recruit TF2H. So TF2H has a really important function in terms of establishing this open complex formation and switching from this initiation to the elongation phase of transcription. And what's important about TF2H is it's a very large protein with a lot of subunits. And it specifically has these two important types of subunits. It has 2 kinase subunits and 2 helicase subunits. So the kinase subunits, if we remember, kinase activity is responsible for transferring phosphate groups. The kinases are going to phosphorylate RNA polymerase 2. So RNA polymerase 2 at the end of it is going to have this carboxy terminal domain, this region of amino acid sequence at the end. And within that, there's 2 different amino acids in it that are both serines, the 2nd and the 5th amino acid of this terminal domain. This kinase subunit of TF2H is going to phosphorylate these serines. And what that does is it kind of activates RNA polymerase 2, and it's going to help kickstart the elongation phase. So when we see this phosphorylation occur, that's going to kind of help denature the DNA and switch to elongation. It's going to help, along with these ATP hydrolysis subunits, it's going to kind of release your TF2B, your TF2E, the TF2H, and that's going to help kind of begin this transcription process where we're not only going to establish the RNA polymerase on the DNA, but we're going to actually start beginning transcribing it and creating RNA. Along with TF2H, we have this protein called mediator. Now, mediator is a co-activator. It's a regulatory protein that helps regulate this kinase activity of TF2H. So depending on whether or not you want to enhance or silence this gene, the mediator can help further phosphorylate or inhibit this phosphorylation, or maybe inhibit the release of these transcription factors that help switch to elongation. What's also important is that if for some reason TF2H is damaged or it loses its kinase function, mediator can phosphorylate the carboxy terminal domain of RNA polymerase 2 on its own, but that's not its normal function. Normally, it acts as a regulatory protein that helps kind of alongside of TF2H. So once we then kickstart to elongation, we have a very simple process where we'll show a DNA sequence in blue. Let's say this is kind of where we're starting. Let's say you have something like G, T, T, A, C, something like that. You'll have RNA polymerase here, which is going to read DNA. And again, transcribe, you're going to be creating RNA in a 5' to 3' direction, just as we did with DNA replication. RNA polymerase is going to recruit, again, a nucleotide triphosphate. So you have a triphosphorylated nucleotide. In this case, we'll have a cytosine. We'll have a cytosine triphosphate. And then RNA polymerase will bind that here to this guanine, which I'll remove the extra drawings. And of course, this cytosine, since it's the first one, remains triphosphorylated for now. But then RNA polymerase will then attract our next one, which will be an adenine. That will lose pyrophosphate. So this will be monophosphorylated, an adenine, uracil instead of thymine, guanine, and then RNA polymerase will just continue along its merry way and keep on elongating in this kind of normal state. So that's how we're starting eukaryotic transcription. We went through initiation and elongation. In the next video, we'll talk about the different models for termination of eukaryotic transcription. So I hope this video was helpful, and we will see you in the next one.